All right, everybody, we're live. There's always this weird kind of moment when I click the go live button before things start happening, so we'll see what that looks like. Anyway, thanks for coming. We got some people in here, which is great. Happy that you joined us. I know it's not just because you want to get 10 bonus points for attending. I know it's because you want to do well in the Keystone, so I appreciate that. Anyway, what are we doing tonight? So, I did this last time. This is the standards for the Keystone. And so the section that we really want to kind of hit on tonight is this chemical basis for life section. So I'm not going to read these all out, but basically what we're getting into here is just how biological macromolecules, right? So what are the four main macromolecules and what do they do? And we want to touch on water a little bit too. Okay, so that's the goal. And we're going to jump right into it because, frankly, there's a lot of stuff to cover here. So hopefully you have a piece of paper or two. My guess is you're going to need at least 2 pieces of paper. And let's go. Everybody can hear all right. I see people typing. Nobody's typing that they can't hear. So let's go. Let's start with water. I don't want to jump in the water too deeply, but just enough. So on this first piece of paper, so we're going to use two, like I said, we're going to use 2 pieces of paper. The first piece of paper, we're going to do water and just dehydration synthesis and hydrolysis. And we're going to set that piece of paper aside, and then we're going to do the 4 macromolecules on the other piece of paper. That's the goal. So let me show you my paper. Here's my paper. So let's start up here at the top, and I'm going to make a note that just says, let me get this where you can see it. Water is polar. And I think that this is really important and something that we should try to remember. So in order to demonstrate this, if we think about water, it's made of oxygen and hydrogen. And we know that oxygen has 6 electrons. And so it's been a long time since we've drawn electrons around something. But I'm going to draw them like that. And we know that oxygen then bonds with 2 hydrogens. So I'm going to draw my 2 hydrogens over here, and we know hydrogens only have 1 electron. So I'm going to draw my 2 electrons for hydrogen. All right, so what does this water is polar thing mean? So what I'm going to do is kind of just come over here and redraw the same thing almost. It's kind of silly. With my 6 electrons for oxygen, I'm going to put my 2 hydrogen electrons there. And I'm going to move hydrogen out just a little bit so this makes sense. And I'm going to put kind of this dotted line around oxygen that kind of encapsulates those electrons for oxygen. Why? Because oxygen is kind of greedy in this case, and the oxygen kind of uses the electrons more than the hydrogen. So they stay around the hydrogen more than they do around they stay around the oxygen more than they do around the oxygen. Boy, it's late at night. Bear with me. So what does that mean? That means oxygen kind of ends up with 2 extra electrons. And if you have 2 extra electrons and electrons are negatively charged, oxygen is going to become kind of a negatively charged element in this case. And since the hydrogens are losing their 1 electron most of the time, they're going to get kind of a positive charge. And so that is what we mean by water is polar, right? That water has a charge to it. And because of that charge, there's some unique properties of water. So one of the things I think you really want to kind of key in on and understand, besides water being polar, is the idea of cohesion and adhesion. So I'm going to write up here at the top just the word hesion. And it's not actually a word, right? It's like kind of the root word. And so what does that word hesion mean, or that root mean? It means to stick. So we're going to stick that up at, no pun intended, we're going to stick that up at the top there just so we can keep that in our memory. And so then we have 2 other things that 2 words that use hesion, the root of hesion. One of them is cohesion, right? So cohesion is a mutual attraction between like molecules. All right. And then I would point out the word co, and I want you to think like cooperate or collaborate, right? So this mutual attraction between 2 like molecules, right? So co and like maybe go together a little bit there. And then the next word that I think is really important for us to keep in mind as we go into the keystones is adhesion. So adhesion is, again, a mutual attraction. And we've seen that word a couple times lately. We saw it when we talked about mutualism in symbiosis when we were talking about ecology. So again, this is a mutual attraction, but now it's between what? Unlike molecules. All right. So this is why when you stick a straw in a glass of water, the water comes up the straw a little bit. Because adhesion, the water is attracting to the straw and it's pulling it up. And this is also why if you have like a glass and you fill it with water and you can get the water to go above the top of the glass, or if you remember the penny lab where you put water on the penny and the water was able to go and form this kind of ball on top of the penny, these two properties, right? Cohesion, water sticking to itself, and adhesion, water sticking to something else. So those two properties are pretty important, I think, and something we should keep in mind, in the fact that water is polar. And that water is polar thing, if you think back to, let me just shuffle through my papers here really quickly. If you think back to last Tuesday when we did this, we talked about how the phospholipid bilayer, right? The phosphate groups at the top are hydrophilic and they were polar, and that's why they stick to water, right? So think back to that. I also do want to point out one thing why I'm thinking about it. So let's give props where props are due. I said multicellular when I should have said unicellular. So Matt, you were right the other night. I went back and looked at it. The paper that I had down for you guys has this, and if I grab my notes that I was working off, it definitely had unicellular. So I messed up, but that's okay. Thanks for pointing that out. There's a lot going on tonight. I'm shuffling a lot of stuff around tonight, so look for mistakes tonight and let me know if you see them. All right? So let's draw a line and let's do something different down below. And this, now we're going to have to draw some molecules, so this could get a little dicey here. The first thing we want to talk about is dehydration synthesis. So let's write that. And it's so hard to talk and write, so I apologize for that. All right, dehydration synthesis. Let's draw two molecules. This is the way we're going to draw molecules here tonight, especially when we're drawing carbohydrates. I think it's a little bit easy. If we draw almost like two carrots, right, and then connect them together, we have a molecule. So we have 6 carbons there. So anywhere two lines meet, there's a carbon. So I'm going to draw two molecules. This molecule plus this molecule. And here my molecules look terrible. And I'm only going to draw one OH on each molecule. Now, you know there's extra hydrogens and OHs on every carbon, but that's okay. We're just doing this, simplifying it a little bit for this. So I wish I had another color pen here. I don't have another color pen, but the art room was kind enough to donate to me a Sharpie that actually works, so that's a good thing. So check this out. If you think back to dehydration synthesis, dehydration means to take water out. Synthesis means to put together, right? So we're going to take a water out. So I'm going to draw one of these little dotted lines again like this. Just to show you, we're taking that H and that HO out. So if we take this molecule and add it to that molecule, what we get is a molecule that looks like this, right? Plus H2O. So plus water coming out. That's dehydration synthesis. So we took water out to put it together. Very important. And so why would we do that? Because that's how we build bigger molecules, right? We have a monomer here and a monomer here, and we can take those together and put them together and make a bigger molecule. And we'll talk more about that a little bit later on. And then the other process that I think is kind of important for us to know on this piece of paper is hydrolysis. There we go. Can't spell and talk at the same time. So hydrolysis. So in hydrolysis, we have the exact opposite, right? We have this molecule that we've put together at some point. Oh, I'm terrible at this. We have this molecule that we've put together. We're going to add water to it. And when we do that, it's going to split that molecule into 2 molecules. And so that would be hydrolysis. Okay. These are just some important things to remember. Why are these 2 things important to remember? I think is a good thing to point out too. They're important to remember because everything we're about to talk about, these organic molecules, are all made of monomers, and all the monomers get stuck together to build bigger molecules, basically. I shouldn't say all. And so what does that mean? They're all put together using dehydration synthesis. And then when we break them apart, they're broken apart using hydrolysis. This is especially important when we talk about carbohydrates, right? So why are carbs so bad? Like everybody's like, eat a low carb diet, eat a low carb diet. Why? Because when you eat a carbohydrate, you're eating a whole bunch of these monomers stuck together through dehydration synthesis. Wow, can't talk. And then what happens is your body starts to break them down. And as your body is breaking them down, what are they doing? They're just splitting them apart into sugar, 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 sugar. All right. So there's the first sheet. That was simple. I have a feeling it gets harder. Let me grab another piece of paper. I need you to do something. So here's me. Here's my piece of paper. Need you to fold it hamburger style. So what we're going to end up with is we have 4 molecules we have to go over. We're going to end up with 1 molecule on each fold. So I'm going to drop this down right here. Let's go back to that paper view and let's get rolling with that because I want to be very cognizant of your time, and it's 8:12 already. So we got 4 to go through. Here we go. First one, carbohydrates. So let's rate carbohydrates at the top. Carbohydrates. First thing that we need to know. Let me see if I can focus that a little better. So the teacher trick. Unfocus it and then bring it back into focus. Even if it's not better, people will think it is. All right, carbohydrates. First trick. First thing we got to write down. What is the monomer? So I hope you're thinking about this in your head. What is the monomer for a carbohydrate? It is a monosaccharide. And there's no way I'll be able to talk and spell that at the same time. It's a monosaccharide. That's our monomer. The nice thing about the monomer for carbohydrates is it says mono in it, right? So that's kind of nice. Next thing we need to know. What's the next important thing to take with us? What is the molecule? So here's where this one gets tricky because you're going to have to draw this molecule in your graphic organizer. So one thing you can think about those models we built earlier this year and what they look like and how you put them together. So the first model you built had 6 carbons in it and an oxygen, but you started by making an oxygen molecule, taking an oxygen molecule and linking it to 5 carbons. One, two, three, four, 5. All right, so we linked it to 5 carbons. And then there was another carbon off of that. And so now all we have to do is go around and add the hydrogens and the OH groups that we need. So every carbon in the ring needs a hydrogen and an OH. So I'm just going to quickly put hydrogen, OH. Hydrogen, OH. Hydrogen, OH. Hydrogen, OH. And then this carbon right here, we need to have an OH, but we also need to have 2 more hydrogens. And I think we need a hydrogen right there too, because we need to have 4 bonds for every carbon. So that is the basic chemical structure of a monosaccharide. So when you're taking the tests, when you're taking the Keystone, think about it for a second. If they're showing you molecules, if you see this oxygen and carbon ring, it's a sugar. It's a monosaccharide. And then you need to take that with you because they could do that. They could just show you a bunch of molecules. You need to understand that ring is a monosaccharide. So what are these composed of? They're composed of carbon, hydrogen, and oxygen. And that's on a 1 to 2 to 1 ratio, which might be important and it might not be really. But we'll put it down. All right, so we have the monomer, monosaccharides. We have the molecule. We have that drawn, what it's composed of. Let's talk about the different types of saccharides we could have. All right, I don't know that this is really appropriate terminology here, but I think it will help you remember it. So I'm going to write across here in big letters, saccharides. Right, so here are the saccharides. So the first one that we should do as our example are the mono, and I'm just going to write mono, so we're going to go monosaccharides, right? Mono, and I'm going to put that equals 1. And so monosaccharides are our simple sugars. All right, there are simple sugars. And there's 3 examples we're going to write down. We're going to write down glucose, fructose, and galactose. There's an A there. And galactose. There's our monosaccharides. The next one we're going to write down are our disaccharides. So I'm just going to put di, so disaccharides, right? Disaccharides. That's 2. That's a double sugar. And so let's put some examples down of double sugars. Those would be things like sucrose, lactose. Maybe you're familiar with lactose because it's found in milk, and some people are lactose intolerant, meaning that they drink milk and get that lactose that makes them sick. And then maltose. Another one we could write down, oligo. That is 3, I think it's 3 to 7, but somebody could correct me on that if somebody wants to look that up. I'm pretty sure it's 3 to 7. Could be 3 to 8. It doesn't really matter. Somewhere above 2 and below like a lot, all right? So oligosaccharides. And I'm not actually going to put any examples down because I don't think they're going to really key in on that. And then the last one we want to put down is the polysaccharides. So anytime we see poly, that equals many. And we will see that word again when we talk about this stuff. So what are our examples of polysaccharides? First, glycogen, starch, and cellulose. Now, here's where it gets kind of tricky. Glycogen is found in animals. Starch and cellulose are found in plants. All right? Good. Let's go above that and do the same brackets, but the brackets are going to go in a different place now. Glycogen and starch are for storage. They store sugar. Cellulose is for structure. So cellulose is for structure. And if you think back to the other night when we were talking about this stuff, as I shuffle papers, let me just throw that down real quick. I know you're trying to copy it. We said over in the cell wall, we had cellulose, right? Cellulose is what builds the cell wall. So, question. I think at the end of each one of these, we want to write a question, and the question's the same one. Where in the cell? Not great grammar, but it will work for what we're doing here. Where is this located in the cell? Where do we find these things? Well, number 1, we find them in photosynthesis, right? So glucose is the product of photosynthesis. It's what we make when photosynthesis, well, we don't make it, but plants do, right? It's what plants make. Where else do we find it? We find it in respiration. It's not the product of respiration, it's the reactant. Right? It's what goes from photosynthesis to the mitochondria so we can break that down and get ATP from it. And then the last place we find these in the cell, well, probably not the last one, but the last one we're going to key in on is in the cell wall, and that would be cellulose. All right. How are we doing? I hope we're doing good. This is heavy, guys. I'm not gonna lie to you. This topic, this chemical basis of life topic, is a heavy topic. There's a lot here, and we're trying to cut through it. Like I said, it took me about an hour and a half last night to kind of figure out what all I think you needed for this. So, let's That's carbohydrates. That's what I think you need to take in with you for carbohydrates. Let's dive a little bit deeper. Go over to the other side of the paper. Up at the top, next molecule, lipids. All right, same kind of flow. What's the monomer? The monomer, well, put a big no here because there is no monomer. This one is different. But I want you after that big no to write, but they kind of pretend like there is. So we need to know the building blocks. And so triglycerides are the one that they seem to key in on all the Keystone information. And so the monomer, even though it's not a monomer because it's not one thing, are 3 fatty acids and 1 glycerol. All right. Next thing we did down below, we're going to do up here again. The molecule. So what is the molecule? We have to draw it. Now this is where it gets kind of tough, right, to draw because this is kind of in depth. First thing we need to draw is the glycerol. So if you remember, a glycerol is 3 carbons. All right, and we need to go around because carbons need to have 4 bonds on them. So I'm going to go around and stick a bunch of hydrogens on it. So here's my monomer, my glycerol. 3 carbons, bunch of hydrogens. Notice I didn't put anything right here because right here is where we're going to connect to the fatty acids. So what's the fatty acid look like? Well, on the end, it has an oxygen bonded to a carbon, double bonded to another oxygen, and then a bond to another carbon. So I'm just going to go ahead and make 3 of those. Right? And so what that is, if you remember when we built these molecules, they look like elephant heads, right? So the last thing we need to do is behind the elephant heads is build a little elephant. So I'm just going to stick 2 carbons behind that. And then all we have to do is drop a bunch of hydrogens. Now, would you have to know how to draw this molecule? No. If you knew how to draw this molecule, does it make it easier to recognize? Well, certainly. But I don't think you're going to have to know how to draw it. I'm running out of room for my hydrogens. That's too bad. It's hard with this marker too. Anyway, there we go. We squeezed them in, which is probably hard for you to read. But we squeezed them in. Now, I forgot one right here. Bam. I'm going to write a little saying over here. I'm going to zoom in on that a little bit. I hope that doesn't mess anybody up. But I'm just going to zoom in on this just a bit. Try and help you out. All right. I'm going to write a little saying here. 3 elephants. Are you saying it to yourself already? Sitting on their fatty acids. Right? Here's the trick, ladies and gentlemen. If they show you a big molecule that looks like this, you look for the 3 carbons, you look for the elephant heads, and if you have that, we have 3 elephants sitting on their fatty acids, we have a lipid. Think about this. So far, you don't really have to memorize how to draw, but you have to memorize how to recognize it. So far, what do we know? If it has a ring with an oxygen at the top, we have a sugar. If it has a glycerol and 3 elephants sitting on their fatty acids, we have a lipid. So this is something you can take with you. All right, so let's put down our examples. So next, examples. Things like fats, waxes, steroids. And then I'm also going to put here phospholipids, right? Because the phospholipid bilayer. And I'm going to draw a little picture here just to remind us that that phospholipid bilayer is a fat. All right. We're doing good, guys. Gals. All right. 2 other things. I mean, we're almost done with lipids. There's 2 other things I think we should write down. We should keep in mind that there are saturated fats, which have no double bonds. And there are unsaturated fats that have double bonds. What are you talking about, Mr. Shooter? It's easy, right? So we take our fatty acid. We have a carbon double bonded to an oxygen, bonded to an OH. Let's make like 3 carbons on there, and we stick a whole bunch of hydrogens around them because carbon always has to have 4 bonds. Remember, that's key. Why is carbon the best molecule for building organic molecules? Because it can make 4 bonds. That's why it can make these big macromolecules. It's that versus this. Same thing. Carbon double bonded to an oxygen, single bonded to OH. Let's add 3 carbons out there. But now the thing that's different is we're going to stick a double bond here. And so we're not going to need as many hydrogens because we have that double bond. Let's see if you can see what I'm talking about. Take this carbon. 1, 2, 3, 4. Each bond, right? So we got rid of that hydrogen and that hydrogen. So this one is saturated. This one is unsaturated because it doesn't have as many hydrogens as it could possibly have. That's really important. All right. Almost there. Where in the cell? Just throwing that bad grammar around all over the place, right? Where in the cell? Well, cell membrane, I think, is the one that really should stand out to us. There's other places, but cell membrane, let's put that down. The phospholipid bilayer. Right? That's where we find it. Sorry. I zoomed in and now you can't see everything. All right, so let me zoom out a little bit. And then I get my hand in front of the light and things go out of focus. And all right. So we're doing good, guys. We're 50% of the way there. We're more than 50% of the way because we did the other one and we did this. We're already at 627. So I'm in a hurry a little bit. Somebody said, you go too fast. I do go too fast, but this recording is here. Stop it when it's over. When it's over, you can go play it again. It might take a minute or two for it to be ready to be played. And then you can fast forward and scrub to, like if you missed something, you can go back and fill it in. But we want to cover the stuff, and I want to talk to you about it. I don't want to rush too much, but on the other hand, I don't want to, whatever. All right, I'm grabbing another piece of paper because that marker bleeds through, and I'd never be able to write on the back. So let me fold it, my hamburger style. You're taking your paper, you're flipping your paper over, and we're moving on to the next one. So in your head right now, you should be asking yourself, all right, we have carbohydrates, we have lipids. What's he going to write at the top of this one? If you said proteins, you are correct. Proteins at the top. First thing, just like we did on the other ones, what is the monomer? And hopefully you're screaming at your computer right now, amino acids, right? Amino acids, because that's what it is. So amino acids. We got to draw the molecule. This molecule is a little bit easier to draw. So we start with a central carbon. How do we recognize an amino acid? Well, it always has a central carbon. That carbon's bonded to a carbon on one side and a nitrogen on the other side, which is one of the ways that we can recognize an amino acid because it's the first one we're talking about that has a nitrogen in it. Good. So the last carbon, double bond oxygen, single bond OH, we see that a lot. The nitrogen is bonded to 2 hydrogens. It has a different amount of electrons. That's why it doesn't need 4 bonds like a carbon. The carbon only has 2 bonds so far, so we're going to stick a hydrogen up here. And then coming off the bottom, we're going to put an R group. So that R group basically means that can change. So every amino acid has this same backbone here. And then the R group can be different. And that could be something as simple as a hydrogen, and it could be something as simple as a carbon with 3 more hydrogens, or it could be something as complex as a big ring, right? But if you're looking for amino acids, if they give you molecules, what do you look for? Look for the central carbon with the carboxyl group over here and the nitrogen over here. If you see that, you got yourself an amino acid. All right, so what is this composed of? It has carbon, hydrogen, oxygen, nitrogen, and I'm going to put a sulfur here in parentheses because that happens sometimes. That can be there. Slide that over for you. All right. So amino acids are bonded together. Let's just write this. Bonded by peptide bonds through dehydration, there it is, synthesis. Same with the carbohydrates we talked about. When we make those big polysaccharides, they're bonded together by dehydration synthesis. But in proteins, they're called peptide bonds. So I'm going to ask you a question. Let's just write the question here. All right? Come on. We'll write it. What's, tired of writing, the first thing what's the first thing? I want dogs. Dogs do when they meet. What is the first thing dogs do when they meet? And if you're saying to yourself, they sniff each other's butts, you're exactly right. And why am I saying this? Oh, it's terrible. Why am I saying this? Because I want you to remember, you don't have to write this, but I'm just going to throw this up real quick. You don't have to write it. But if we take an amino acid, nitrogen, carbon with its hydrogen and its R group, to another carbon with its hydroxyl group, and then over here we write another amino acid, so H going to a nitrogen, to a carbon, carboxyl, got an H up here and an R group here. When these things bond, I'm sorry, there should be an OH there. When these things bond, no, not at the double bond, I'm sorry, there should be an OH here. Right here is where we get our water. So the bond actually ends up happening between a carbon and a nitrogen, right? So if you remember when we built this, it looked like the head of a dog here, and this would be the tail. This would be the back end of the dog. All right, just throwing that out there for you to remember. That was a little extra for you kids tonight. All right, next thing we need to write, polypeptide. Oh no, we made a serious error here. Polypeptide chains fold into proteins. Right? So polypeptide chains fold. So we get a polypeptide chain, and then it folds up to make the protein. What's important? Shape. We're almost done with proteins. Shape is important. It's three-dimensional. Does this ring a bell? We have an enzyme. Here's my enzyme. Let's label this enzyme. In the enzyme, we have a substrate. Right? That enzyme breaks the substrate. So the enzyme doesn't disappear. Remember, they can be used over and over again. But the substrate, because it was in the enzyme, breaks in half. Keep in mind, shape is important because if we change the shape, the substrate doesn't fit anymore. So the big question here is, how do we mess up a enzyme? All right, we could do semi-proper language. How do we mess up an enzyme? We do that with heat and pH. So do you remember all that? Heat and pH will change the enzyme's shape. All right? Let's keep going. We're almost there. So where do we find this in the cell? Where in the cell? My hand is cramping. How are you guys doing? Oh, it's a good thing I can't hear you complain. All right, where in the cell? Well, we would find enzymes at the ribosome, because the ribosome is taking tRNA and mRNA and making enzymes. We would find them probably floating around in the ER because they were made in the ribosomes. It's on the ER. And we would also find them in the cell membrane. Cool? All right. We're on to the last one. And this is the shortest one simply because we're going to talk about this more in another one when we talk about DNA specifically. But for tonight, let's get a couple things down. Nucleic acids. So nucleic acids. All right. What is the monomer? It is a nucleotide. What is the molecule? This is the one I'm dreading. What is the molecule? Well, the molecule is big because it's made of a whole bunch of stuff. And I actually have it drawn out here. But you know what? I think we're going to skip that. We're going to draw it differently than I have it drawn out with all the carbons and the phosphates and everything. Let's draw it like this. What if I draw this? Does that ring a bell? Because all the models you cut out looked like this. Let's label it. So what do we have right here? This is our sugar. This is our base. And this is our acid. So a nucleotide is made of 3 parts: an acid, a sugar, and a base. What are our examples? We have DNA, RNA, and ATP. I want to put ATP in there because that one kind of stumped us earlier in the year that was actually a nucleotide, nucleic acid. All right, quick chart. We're going to list the different types of nucleic acids over here. I'm just going to put Na for the nucleic acids, and then here we're going to put the bases that they have. So if we are looking at DNA, it has an adenine, thymine, cytosine, and guanine. If we are looking at mRNA, tRNA, or rRNA, we have an adenine, no thymine, uracil, cytosine, and guanine. We could also write that these are double-stranded, and these are single-stranded. All right? And that's really what we need to cover. I think we got to ask our kind of grammatically incorrect, where in the cell, are these located? So let's say they are first in the nucleus. Right? So what's in the nucleus? DNA. And then parentheses write chromatin and chromosomes. Where else are they located? They're in the ribosomes. So what's in the ribosomes? That is rRNA. Oh my gosh, rRNA. But we also, because ribosomes are made of rRNA, we also have tRNA there. We also have mRNA there. And we'd also find this in ATP, right? I say energy, you say what? I hope you screamed that out for your parents. All right, it took me a little longer than I wanted to, but hopefully you stuck with me. I'll tell you what, I think if you stuck with me, and it looks like a lot of you did stick with me, I really appreciate that. So what I'm going to do is your last job here tonight. Show me these graphic organizers. You get your 10 bonus points tomorrow. But I'm going to give you 5 extra for sticking with this the whole time. I need you in the chat room just to type your name and period number. Put your first name and period number or something like that. Stick that in the chat room real quick. And when I hang up, I'll pull up the grade book and add 5 bonus points. I'm going to put all the bonus points in that same column. It's like the first column in the grade book. So if you have 10 from Tuesday, it will go up to 15. And then when you show me your graphic organizer, it will go up to 25. So before you hang and get out of here, stick in your name, period number. I'll put an extra 5 bonus points in there for hanging with me on this long one tonight. Because this one was rough, right? There's a lot of writing and stuff. I'm done with Cap. I'm done with the content. Jackson, way to go, man. Stick around. Jackson, stick your period number there, and I'll give you the bonus points for it. All right. Give me a name, and I'll do that as soon as I hang up on this. I don't think the next two will be quite this big. This is the hardest one, guys. But if you think about it, this one is kind of the foundation for every other thing we're talking about. Right? So if you really have a good handle on the organic molecules, this stuff, if you have it down, that's going to make so much of this test easier for you. All right? So stick your name, period number there in the comments. Like I said, as soon as I hang this up, I will throw that in for you. I appreciate you coming. I appreciate you sticking around. I appreciate that you guys want to do a good job on this test. I want you guys to knock it out of the park. I don't want you wasting time in the future. I don't want you sucked into remediation and things like that. So I really am glad that you're around. I'm looking over here. Is there any questions? Thanks, Mr. Shooter. You're very welcome. Nina loves writing. I love writing too. Song tonight. No, there's no song tonight. I'm going to make you wait for it. And I don't know what song you'd want me to play anyway. Song. Boy, that would just be terrible. That really would be. You get a nice view of the paper. All right, like I said, it is 8:41. We're 10 minutes past what I wanted to do. I love giving up my time for you, but I've held you long enough, so there's no way I want to bore you with another song. So you got your comment in there, your period number. I see that. I really appreciate that. I will see you tomorrow. Hey, ladies and gentlemen, tomorrow is Friday. Tomorrow's Friday. We got one more week. We'll talk about evolution. I will see you then. We Didn't Start the Fire by Billy Joel. Sorry. I am not the piano man. All right. Have a wonderful night, everybody. I guess by next Thursday, I'm going to really have to think about this song thing. Maybe I'll fire up that Marshall amp sitting back there behind me. Can you see that over my shoulder? Right here. No. Right here. This Marshall amp. That's the loud one. All right. Have a good night. See you tomorrow.